Gil McCrae is going to come forward and get our red tide discussion going here. Thank you, Gil.
Thank you, Mr. Chairman. Good morning, Commissioners. This will be a dual presentation by myself and Jessica, so you will see us transition at some point during the presentation. Just a couple caveats before I start. I think you all know that if you look at the updates on our website that this is an ongoing event. We all hope that we're near the end of the event, but the information I'm going to be presenting today is as recent as we have, and keep in mind that some of the numbers, especially relative to fish and wildlife impacts, will change. So here's the outline for the presentation. I'm going to essentially do a quick Red Tide 101. We'll talk about ongoing efforts relative to funding control and mitigation activities and highlight our partners both within and outside government. I think it's important for you all to realize that an event of this scale and magnitude takes a lot of different players working together, and as Eric said, that has gone really well up to now. I'll give you an overview of the current red tide, including tracking it from its beginning in November of last year to where we are today, and then talk a little bit about the fish and wildlife impacts, because that's one of the things that we focus our work on as FWC, tracking those impacts and determining what the long-term repercussions might be, or if management action is warranted. And then Jessica will cover the management side, and she'll have a staff recommendation at the end of her presentation. That is a photo that was put in by someone who wasn't me. So I think that is probably a combination of red tide and tricho. Do you see it in there? Yeah, Kate says it's red tide, and I believe her.
Next slide.
Mr. Chairman, would you like to have the controller? I've almost gotten the hook before, but I've never had to give up the controller. In all seriousness, we hear about harmful algal blooms all the time, and we forget that the marine algae, phytoplankton, without them, we would not be on this planet. They're an essential component of marine food webs. Like all plants that photosynthesize, they produce oxygen, and because of the sheer magnitude of our oceans, marine phytoplankton, it's estimated, produce about half of all the oxygen we breathe. So when we talk about harmful algal blooms, it's really something that's essential to life on Earth, proliferating and creating problems. But algae itself in the marine environment is critical to marine food webs and all life on Earth. Now, harmful algal blooms can have two components that cause trouble. Certain algae produce toxins which can impact fish and wildlife either directly or indirectly. Red tide does that. Or all blooms, and really all accumulations of organic matter that's outside the norm, when they decay in aquatic systems, they can create low dissolved oxygen conditions because that decomposition process eats up oxygen. So any bloom, whether it's toxic or not, can create issues as the bloom decays and eats up oxygen. So what is red tide? Red tide is a particular form of marine algae. It's in a group called dinoflagellates. It produces a toxin, and this toxin actually has several different forms, but as a group they're known as brevetoxin. The scientific name of red tide is Karenia brevis, so you may hear me fall into that during this presentation. That is red tide. This toxin can kill fish, birds, sea turtles, manatees, dolphins directly or indirectly through their diet, which is the most common exposure path for everything but fish. Fish it can kill directly. Now, most invertebrates, except for scallops, and I'll touch on that in a minute, aren't killed by red tide, but the bivalves you see here, the clams, the oysters, their mussels, they're filter feeders, and they accumulate the algae in their system, and as they accumulate the algae in their system, they become unsafe for humans to eat. In fact, if humans eat a clam, a mussel, or an oyster that has red tide, it can cause NSP, neurotoxic shellfish poisoning, and that's a very serious threat to human health. So that's why FWC works really closely with the Department of Agriculture to close shellfish harvesting areas when red tide is in the area. So all of you have heard about this before, but one of the most important interactions we have with our partner agencies is that seafood safety interaction. Scallops are a little different. They are the only bivalve that actually is killed directly by red tide. So what you heard about in St. Joe Bay is a concern from a human health standpoint because Dax has closed the bay due to red tide, but also a concern from the scallop population because scallops can be killed directly by red tide. And I think most of you know that if you're on the beach during a red tide, you will get this kind of acrid smell. That is the actual red tide toxin being carried in the air in the sea spray. Usually not a problem for folks who don't have asthma or other respiratory ailments, but it is irritating and it does make your day on the beach unpleasant. That is the red tide toxin carried in aerosol. And then of course we all know about large fish kills that red tide can cause. Not only a concern from a fishery standpoint, which typically the populations are fairly resilient to, but also economically and the cleanup efforts that most of all the counties have shouldered the burden for those. That is the red tide organism there in the lower left. It is not visible to the naked eye. The chairman was out at our lab last week, I think it was, and he got a close-up view of these guys. They're a very unique organism. In fact, they have two flagella, the little strings, I guess, that help them swim. And because there's two, they have a unique swimming pattern, kind of a corkscrew pattern. And there's actually a way that we take advantage of that swimming pattern using software and artificial intelligence to use a package that identifies them on a microscope slide based on how they swim. So much of what I'll be talking about today relative to the state of the science is the technology keeping up with the reality. Red tides have been around for a long time, but only in the last 10 or 20 years have some of these tools become available for us to understand things we couldn't understand before. So a little bit about red tide history. I'll caveat this by saying that it's only been about in the last 30, 35 years where we've had satellites capable of picking up red tide offshore. The satellites are simply picking up the photosynthetic pigment in the algae. So it's reflected light, and of course, if that satellite hits cloud cover, you don't have information. So the satellite information is dependent on having a clear day. But the reason I say this is our long-term record is incomplete because prior to about the late 70s, we only found out about red tide if it came inshore and caused impacts, or if fishermen typically reported fish kills offshore. So our record is incomplete. But you can see that we have had red tides. You'll see in a future slide, red tide occurs almost every year. The oldest reports we have anecdotally are actually from the native cultures in Florida, the Calusa in particular in southwest Florida. The Spanish reported on their oral history. Those natives had no written language, unfortunately, otherwise we might have a pretty significant record to refer to, but it's been around for hundreds of years. And prolonged red tides are less frequent. Recognizing the caveat that I gave earlier about technology picking them up only in recent years, you can see the list here of years where we had prolonged red tides. And this year, thus far, we have an 11-month red tide you see in 17-18. Now, we have been, in our researching of this event, we've gathered some records from a red tide in 1946 and 47 that you see here as well. It was an 11-month red tide. Also started in November of 1946. So there's some parallels with the red tide we're having right now and the very significant red tide that happened in 1946 and 1947. Just an interesting anecdote, this was right after World War II, and back then a large segment of the public were convinced in 1946 that the red tide was caused by munitions dumping in the Gulf after World War II. So that was a theory that was quickly debunked even then. So a little bit about the two different algal blooms that you've heard about recently. Eric mentioned this and Secretary Bartlett mentioned this and did a good job differentiating the two, but a little bit more information on this. First of all, red tide starts offshore in the Gulf, in fact 10 to 40 miles offshore, and there's a particular initiation zone that is of interest to scientists, and I'll talk about that in a minute. It is a saltwater algal species. It cannot live in fresh water, so it occurs in estuaries, coasts, and offshore in the Gulf of Mexico. It produces brevetoxins, as I mentioned, and you see the impacts here that I mentioned earlier. Now, on the other hand, blue-green algae, especially the one that we deal with most frequently in Florida, microcystis is its name. It can produce a toxin, microcystin, but it doesn't always. And this is one of the confounding factors with researching these algal blooms. Sometimes you'll have an algal bloom that produces toxins or less toxins. Sometimes it produces more, and we don't always understand why. Now, microcystis typically does not create kills directly. Its main impact on fish and wildlife is through that low dissolved oxygen as the bloom decays. Now, it can also create problems for drinking water, but in Florida, I think it is still the case that the majority of our communities draw drinking water from groundwater. So the drinking water issues with blue-green algal blooms are a big problem in other parts of the country, but I believe not as big a problem or a concern in Florida. The big thing to realize is that these blooms do not occur together. Freshwater kills red tide, and saltwater is not good for blue-green algae. So getting back to red tide and the way it functions in the ecosystem, it is one of the most ecologically flexible organisms that we study. Even though it can't live in fresh water, it does have a very broad salinity and temperature tolerance. And more importantly, it's capable of using a lot of different nutrient sources for growth. And this is the question we get most frequently, where are the nutrients coming from that fuel this red tide? The first thing to realize is that these red tides start offshore in an area that naturally is very low in nutrients. So this is where this diverse use of nutrients comes into play. There's a few of them mentioned here. The most significant one, and one of the more interesting ones, is there's a connection between red tide and another marine blue-green algae, which is non-toxic, called Trichodesmium, and you see that referenced here. Trichodesmium can take nitrogen out of the air. 80% of the air we breathe is nitrogen. I think I got that percentage right. And it converts it into a form that's biologically available for red tide. So one of the things we look for when we're researching red tides is whether or not Trichodesmium blooms are there, because if they are there, we know there's a mechanism for this other algae to convert nitrogen into a form that red tide uses. And I'll touch on this a little bit more, but that is increasingly becoming a significant area of research for harmful algal bloom scientists. Now, red tide kills a lot of fish, and those dead fish also generate nutrients that red tide can use. So all of these are in addition to the nutrients that occur naturally, the nutrients that are in the ecosystem, the nutrients that are typically higher in inshore systems. So one of the tough things about researching red tide is there's often no way to parse out these categories of nutrients and which category is more important for an individual red tide versus another. So back to occurrence rates. This is the number of years that, on the map on the right, the number of years that red tide has occurred since 1953. That's 66 years. So you can see in Southwest Florida, 57 of those 66 years, we had a red tide somewhere. And again, we may have missed a few years early on, especially when we didn't have the satellite technology. This is why we think the initiation zone is offshore of Southwest Florida. It makes perfect sense. Now, most commonly in the Gulf, other than Southwest Florida, red tides will go up into the Panhandle or down south. In fact, with this current red tide, we have had hits both down south and up in the Panhandle. It's much rarer for red tide to go to our East Coast because in order for that to happen, it has to be transported out of the Gulf. So red tide is a Gulf of Mexico occurrence. For it to show up on the East Coast, it's got to get caught up in the Florida current and the jet stream and make its way to the East Coast. So this is the current thinking relative to how red tides start. And there's a lot of science that goes into this simple cartoon, but in a nutshell, during certain parts of the year, typically fall, which is the most common red tide initiation seasons, the combination of surface winds blowing offshore and ocean currents out of the southwest create upwelling conditions. And these upwelling conditions do two things. They bring nutrients from the bottom of the ocean up to the surface, and they also bring red tide cells from the bottom to the surface. And this is where technology like gliders comes in handy, because if red tide is at the bottom, the satellites will not pick them up. clearly. They're hundreds of feet down. So that glider technology can get down there. It can find a signal for that chlorophyll. It can direct our samplers to go out and take bottom water samples and document that that red tide is on the bottom. And in this current event, we've actually been able to do that, and I'll talk about that in a minute. So once these patterns of surface winds and southwesterly currents create the upwelling, bring the red tide up to the surface, it goes into its diverse nutrient use mode, and it pulls nutrients from wherever it can get it. And the inshore impacts that we've seen in this particular red tide are mainly due to patterns of winds and currents that have moved it inshore and kept it inshore, which is unique to this event. So given all that background, I just want to highlight a few ongoing efforts relative to the whole area of control and mitigation. I think when you look at a satellite map, when you look at photos like this, which I believe this is in Pinellas County from this year's red tide, it's a very daunting task to try to control something like red tide. But there are situations, especially in residential canals or even with aquaculture operations where you're protecting your product, where control of red tide might be feasible and something you want to do. So this is where the governor's funding recently has targeted. There's work going on with clay. It's a new formulation of clay that has not been tried in Florida before, ozone, and a couple other different techniques. So this is a funding vehicle that is going to get all of that work going very soon to see what sort of success we might have in mitigating the effects of red tide. I would be remiss if I didn't point out our partners at FWC cannot do this alone, even though we are the point agency for red tide. We rely on a number of partners, and I'll just go over their roles very quickly. So in FWC, we are the monitoring center. We produce the red tide status reports twice a week on our website. So today is Wednesday, I think. So there will be a report today, and there will be another report Friday. So on our website, myfwc.com/research, you can go out there and get the latest on what the red tide is doing. And these are statewide samples, and I'll show some examples in a minute. Secretary Bartlett talked about DEP. They've been a very valuable partner in this effort. They do water quality monitoring. They are very valuable in looking at these control and mitigation techniques. And of course, all of you know that the millions of dollars that have come to counties for cleanup, which I know the counties are very appreciative of, DEP has helped manage that process. And it's really taken a load off our backs with DEP helping with that funding, because if you're at the county level and you're trying to deal with the fish kills, it's a very challenging exercise. I mentioned DACS earlier. They are the seafood safety agency in the state. For decades, we've had a partnership with them. Formally, it's called the Biotoxin Contingency Plan. And I'm happy to say that Florida has not had an illness from a red tide contaminated shellfish in decades. This is a very successful program, very conservative, and it keeps Florida's seafood safe. The Florida Department of Health deals with all of the human health issues. They have been very active. The Surgeon General was at the meeting with Eric and I at Moat just last week, I think, or was it this week? Monday. Today is Wednesday, for the record. I had a chance to chat with her for quite a while, and she's very interested and engaged on this issue, and she wants to know more about how red tide might affect human health for coastal residents and our visitors. So they're very valuable partners as well. And on the science side, you heard Moat Marine Laboratories name quite a bit. They are a key partner. We've had a cooperative red tide research and monitoring program with them for decades, and they are taking the lead on certain control and mitigation options, especially the ozone. NOAA, our federal partner agency, has an harmful algal bloom operational forecast system, which is geared toward beaches and respiratory irritation on beaches. That creates a public outreach component that is very helpful when someone calls us and wants to know whether a certain beach is going to be pleasant on a particular day. We have some place to point them. And then the University of South Florida College of Marine Science is also a critical partner. In St. Pete, they're right next door. Our buildings are literally connected with a bridge. They provide the satellite imagery that we rely on more than people realize. We depend a lot on satellite imagery, and they do the forecasts. Right now, we're able to forecast movement of red tide in a 3.5-day time period. So USF College of Marine Science valuable partner, minute to minute, joined at the hip on red tide. So what I'm going to do now is I'm just going to walk you through the history on the current red tide, and as I do, I'm going to try to point out some trends that are characteristic of red tides and how this red tide differed a little bit. So the first few series of maps you'll see will be monthly aggregated averages of red tide concentrations, and I'll just point out a couple things with this map. Every dot you see here is a water sample. Even the gray ones, that means that someone has gone out there, collected water, gotten it to somebody who looked at it under a microscope, determined if there was red tide, and if there's red tide, what the concentrations were. And the colors here represent the concentrations of red tide. So anything orange and red is over 100,000 cells per liter. That's considered a bloom condition. So if you see orange and red, you're in a full-blown red tide bloom condition. The yellow and white are lower levels of red tide, and keep in mind that red tide occurs at background levels naturally in our coastal waters. So in October of last year, we saw the first hits of low concentrations of red tide in Southwest Florida. By November, just that next month, we had gotten bloom conditions in several counties. Now, this is the normal season for red tide. Red tide is typically a fall phenomenon, and the standard process that happens is red tide, if we have one, they'll begin in the fall, but they peter out in the wintertime, and certainly by spring. This one did not do that. So this was November. The concentrations stayed relatively high but spotty through December. And this is the time period in January, February, March where we would expect things to dissipate a little bit traditionally. We still had bloom conditions in Southwest Florida. And you can see all the dots that are gray, the sampling is continuing to go on. Same thing in February. This is a little bit atypical because those bloom conditions now are persisting when we would expect them to dissipate. This is about the time where the northern fronts that come down to Florida stopped this particular winter. Normally those fronts would continue into March and they'd keep the red tide offshore. So part of what we're dealing with this year is an anomalous weather pattern where those fronts coming down from the north stopped in February. So going into March now, we don't have those fronts pushing the red tide offshore, and we're getting some stronger hits of high concentrations in Southwest in March, which is, it's atypical. Sticking around in April, a little back off in May, but back again in June. All right? So you can see by this time the counties in Southwest Florida have already been dealing with some level of red tide for several months. Now I'm going to go to a weekly map here, and I'll go through this really quickly, but you'll see the progression from June, July, and through September. And keep an eye on that area of Southwest Florida, Sarasota, Charlotte, and Lee County. So that was early July, late June, early July, going into July, mid-July, late July, pretty high concentrations inshore. And this was also unusual because the current and wind patterns, which typically move some of the red tide offshore in a normal year, we're keeping it inshore. So again, the big difference with this red tide is long residence time inshore. So that was late July, going into early August, very similar pattern, expanding a little bit north and south into August, mid-August, late August. A little bit more patchy here in late August, into September, and through to where we are today. That's the report as of last week. Now I can tell you that it is Wednesday again, so the report that will come out today will show a little bit more patchiness in Southwest Florida, which is a good sign. We think that's probably due to winds coming out of the east and the north. So hopefully that trend will continue because that really mitigates some of those inshore impacts. All right, I'm just going to talk very briefly. I mentioned a little bit about how we monitor and track red tide. Really, there are three main techniques. The satellite monitoring, which picks up the chlorophyll, but does not work well inshore because there's too much backscatter. Remember, we're looking at reflected light with a satellite. We have a limited but very valuable ability to get essentially underwater robots offshore, and these are the gliders. These gliders will contain sensors that allow us to understand what's happening not only at the surface, but also mid-water column and on the bottom. And then the standard tried and true method of going out and collecting water samples, getting them to a scientist who looks at them under the microscope. The map you see in the upper right there is an example of some of the forecasting we do. And again, our forecasting methodology is limited to about 3.5 days in terms of where the red tide's going to go. So I wanted to give these ladies a shout out because they are in our lab in St. Pete, and Eric and the chairman got to meet a few of them, and this number has gone up now to over 10,000 water samples. Those dots on the map that I showed you over the months and months, the bulk of those samples come into this individual room where this team of scientists generates that information that populates those red tide status reports. And they've done a phenomenal job. I made a point of going down late every day and checking on them and make sure they were holding it together, and they're holding it together better than I am. So I don't have to go down as much anymore. But the photo on the right is a very recent photo from Reddington Beach, which is in Pinellas County, and it shows you how different some of these concentrations can be. These are both really high concentrations, bloom concentrations, but just a few yards away you go from 18 million cells per liter to 51 million cells per liter. And if you get a sample that has 51 million cells per liter under a microscope, it is just layers of red tide that these women painstakingly work through and generate that data. So very appreciative for their efforts. So this is the glider, and this takes a little bit of explaining. It's essentially an underwater robot, and it's programmed to follow a certain course, and typically it yo-yos through the water column. And I want to point out, because this is a very recent glider mission, late October, early September, and this is the track that it ran, and it's yo-yoing up and down the water column. For reference, this is Tampa Bay right here, and again, this is University of South Florida College of Marine Science. So this is the chlorophyll signature from that particular mission, and you can see, or maybe not see, it's late August through early September. And what I want to point out are these hits on the bottom. These hits on the bottom are chlorophyll, and what that means is that's concentrations of algae. And if you look at this profile, you don't see those hits at the surface. So the satellite wouldn't have picked this up. A fisherman running a charter trip taking a surface sample wouldn't have picked this up. And this essentially is what we think is happening with red tide. There's concentrations of these algal blooms on the bottom that are then upwelled under certain condition to the surface, and that's when the bloom condition occurs. That's a very simplified version, and if Kate's making a face right now, it's probably with good measure, but that's basically what the gliders can give us. Now, was she making a face? Okay, good. What the gliders can give us is the chlorophyll, but in order to determine that that's red tide, we have to have the glider park there, get a boat out there, and take the water sample at depth. And not on this mission, but on another mission, we actually did that and confirmed that some of these chlorophyll hits we're getting on the bottom were in fact red tide. So that's just another monitoring technique that bolsters the theory that I spoke about earlier. The other thing we're doing this year that is a little bit unique given the scope and scale of the event is weekly aerial surveys. And this is where we've really relied on our law enforcement aviation group, who's been a tremendous resource. And this is just an example of transects that we flew in September. And you can see what it gives us is it gives us this large-scale view of the patchiness of the red tide. It also helps us pick up the other algal bloom Trichodesmium, which I'll talk about in a minute. But this is one more high level of resolution information to go with the water sample. So this is an example of an aerial survey photo that we had on 9/6, and I'll point out a couple things here. This is actually a situation where we saw the two algae side by side. The whitish algae is the Trichodesmium. Remember, this is the one that can take nitrogen out of the air and make it available for red tide. And the brownish algae here is red tide. So we have numerous examples of these blooms co-occurring, and it just reinforces again and bolsters the theory that there's a connection between these two types of blooms. The other thing I want to point out is that the The nitrogen that, or I'm sorry, a key nutrient for Trichodesmium is iron. And one of the largest sources of iron to offshore ecosystems is from the atmosphere. And desert dust, particularly dust from the Sahara, contains significant amounts of iron. Keep in mind the Sahara is as big as the continental United States. So when there's a dust storm coming from the Sahara, this time of year, the winds are generally moving, in fact, actually all times of year, but in particular this times of year, the wind's taking the dust from the Sahara toward us in the Caribbean. And in fact, we know from NASA satellite imagery that in June of 2018, there was a significant dust event that brought a lot of Saharan dust and therefore also brought a lot of iron. On an average year, the hundreds of millions of tons of Saharan dust comes our way. This photo from June 2018 was about 15 times the long-term average for Saharan dust. So sometimes when we talk about Saharan dust, people think we've gone a little bit loopy, but the connection between iron, trichodesmium, and red tide makes logical sense. In this particular year, it's been an unusually high year for dust deposition. Okay, so now we'll get a little bit closer to home and we'll talk about fish and wildlife impacts. So reiterating a little bit, red tide can kill fish directly, or it can impact other animals, other wildlife, through their diet. And diet is the main exposure pathway for things like manatees, sea turtles, bottlenose dolphins. Red tide can also alter behavior and distribution patterns for fisheries, and many of our fisheries go to certain places at certain times of year to spawn. And so alteration of those patterns can affect productivity of the stock. The thing to keep in mind is that red tide can be very patchy. Sometimes you'll get lucky and it will not be in a place that's a critical life history staging area for fisheries. Sometimes it does cause problems. So one of the things we'll talk about, especially relative to fisheries, is the timing aspect and the patchiness. And again, this bloom has persisted inshore longer than past blooms. So a little bit on wildlife impacts. Here we're mainly looking at, excuse me for a second, we're mainly looking at sea turtles, manatees, and dolphins. Reiterating the fact that the primary exposure is through diet. So on sea turtles, we've had 3 species that have been the bulk of the impacts from red tide: loggerhead, Kemp's ridley, and green sea turtles. Loggerhead and Kemp's ridley are carnivores. They eat bottom-dwelling organisms, clams, shrimp, invertebrates with hard shells. This is why loggerheads can eat them. They have this giant head with a lot of crushing power. So that is why those 2 species, loggerheads and Kemp ridleys, make up the bulk of the sea turtles that have stranded. Now, we use the term stranding with sea turtles for both dead sea turtles and turtles that have been incapacitated but alive. Some of these turtles can be saved if they're gotten to in time and taken to rescue or rehab facilities. So that dietary exposure is the mechanism that is affecting loggerheads and Kemps. Green turtles are a little bit different. There's a lot of green turtles that nest in Florida, but they're a vegetarian. So their diet is less of an exposure pathway for something like red tide, and lower numbers of green turtles have been affected. To date, about 460 turtles have stranded due to this event. Just to put that in context, last year Florida had close to 100,000 loggerhead nests and close to 50,000 green turtle nests. Kemp's ridleys nest in Mexico, so we don't have as good a data for Kemp's ridleys. The thing to know about sea turtles is relative to nesting, and this bloom occurred at the end of their nesting season. The females that nest aren't actively feeding during that time, so they're not being exposed to the red tide. So that helps with this year. Sea turtles will nest only every 2 to 3 years, so it is possible that sea turtles were killed this year that would nest in subsequent years, and that's something we'll keep an eye on. But again, with 100,000 loggerhead nests in Florida, 50,000 green turtle nests in Florida, there's a large capacity to compensate for that level of mortality. If you're wondering about the hatchlings, they are emerging from their nests and they are heading offshore as quickly as they can, because that's what the hatchlings do. They have residual yolks when they hatch out, so they do not actively feed until they get offshore and typically get into those sargassum and seaweed beds where they hang out. So in general, the hatchlings do fairly well, even if there is a red tide on their nesting beach. Manatees, we've picked up about 160 manatees as part of this event, ranging from Manatee to Collier counties. That's the second highest number we've seen with manatee mortalities. In 2013, that was the worst year we had for manatee mortality. We had about 277 this year. Relative to long-term impacts for manatees, the modeling that we used to project the future population status of the manatee and the modeling that contributed to the federal downlisting from threatened to endangered, it incorporates a red tide element in it, a certain probability of a severe red tide regionally. So Southwest has a higher frequency than, say, Northwest or the East Coast. Depending on the data that we get from this particular event, those models will be adjusted and account for that. But in terms of long-term impacts, that modeling already considers red tide. So I don't think we'll have any surprises there relative to the level of mortality that we've seen. About 60 bottlenose dolphins have stranded. NOAA is the lead on that particular area. That data is still being compiled. The long-term implications of that are still unclear. And again, the event is ongoing. One of the big bump ups that we had that had real on-the-ground impact was the extra funding to MOTE for marine mammal response. You can imagine that our folks that go out and rescue and respond to marine mammal carcasses are running pretty ragged these days, and it's my responsibility to make sure they continue to do that safely. We have people that are very passionate and will put themselves in situations they shouldn't put themselves in because of that passion. I have a passion meter I need to check on periodically, but this extra help puts more bodies into the effort and makes my job a little easier. And then that funding also was used to do a rapid assessment, which was led by MOTE Marine Laboratory, of the Snook populations in Southwest Florida. So they got on the water very quickly to look at the Snook populations. A little bit more positive side, both with sea turtles and manatees, we can rescue them, and they do pretty well in rehab facilities if we get to them in time. And so far, 12 manatees and upwards of 50 sea turtles have been rescued. Also, talking a little bit about fish kills. I mean, this is the most significant manifestation of red tides. FWC operates a fish kill hotline. We've had well over 1,200 calls to this hotline during this event. At some point, we imagine people stop calling because they assume the event has been reported. So probably a lot more than that if people had reported everything. More than 100 species affected by red tide, and again, hundreds of tons of fish cleaned up primarily by the counties, who I know are very grateful for the extra support. Millions of dollars go into the counties to help them clean up fish killed by red tide. Okay, so the next series of slides, which I'll try to go through fairly quickly because I know I'm going on a bit here, deal with impacts to two of our iconic inshore game fish species, snook and red drum. And I'll begin the discussion by pointing out that in Tampa Bay and Charlotte Harbor, we've been doing scientific surveys for these species for over 20 years. So we have a good baseline of what abundances of these fish should be in those systems naturally. So if you think about it, and we're wondering whether or not we have impacts or whether or not we need to think about whether regulations can go back to what they used to be if in fact we have more severe regulations, we're looking at that baseline of 20 years to refer to. So as I walk through slides that are specific to these two species, I want to point out that the reproductive characteristics of the species, the timing issue, when they spawn, where the juveniles end up, and our past experience with red tides, it informs what we think about how red tide is affecting these particular species. So let's look at snook first. You might remember that snook changed from male to female, which is backwards from what a lot of the groupers do. It's hard to keep track of, I know. They do mature early, they live fairly long, and that creates a moderate resilience to events like red tide because they can turn over their population pretty quickly. They spawn a little bit earlier than some of our other species, so they had by the time the red tide had gotten really bad in June into July, they had a few months of spawning already. So that is helpful in terms of resilience. The other thing that's helpful with snook is they will use freshwater habitats, both as adults and juveniles. So because red tide can't live in freshwater, those habitats can provide refuge for these snook. All right, now this is going back to the 20-year history that I mentioned earlier. The table that I have here compares the abundance of snook that we saw in July and August of this year from our sampling to the 20-year period. So if it says comparable, for example, adults in Tampa Bay, our monitoring shows that the abundance in July and August of 2018 was comparable to that long-term average. So this boils down a lot of information into a very simple table. And you can see here that because of this timing of reproduction and this ability to use freshwater as a refuge, in general, both adults and juvenile, up to August 2018, are doing fairly well both in Tampa Bay and Charlotte Harbor. However, keep in mind the event is not over and the impacts are far from being fully understood. Going on to red drum, the situation is a little bit different, a little bit more complicated with red drum. They're a little bit less resilient than snook. Those of you who fish for red drum inshore are fishing on young red drum because they only stay in those inshore habitats till about age 3. After age 3, they move offshore and they never come back. So when they move offshore, the next 40, 50 years of their life, there are these big bull red drum that you might hear about. They're way out of the slot, so they're not caught in state waters. And the timing of the reproduction for red drum is fall. So right now, actually, these bull red drum that are living offshore are aggregating in these very large aggregations and just beginning their spawning behavior. So that timing is not ideal relative to the red tide, and I'll talk about that a little bit more in a minute. The story becomes a little bit more complicated when we look at the July and August abundances compared to the long-term average abundance. The reason it says larger juveniles and younger juveniles here is because inshore they're all juveniles. So that can be a little confusing. It has more to do with the different gears we use to catch large versus small red drums. And you can see that the juvenile abundance in July, in July through August 2018 was below the long-term average for both Tampa Bay and Charlotte Harbor. We don't think this is due to red tide because we picked up a signal a few years ago where we had 2 bad recruiting classes that resulted in lower juvenile abundance of red drum in these estuaries. Keep in mind, these are only age 3 fish and below in the estuary. So if you have 2 bad recruiting years within a 3-year period, you're going to have low abundance of red drum inshore. So what this is saying is that any red tide impacts to red drum inshore are affecting a population that is already at abundance below the long-term average. Just a little bit of information on red drum spawning aggregations. You recall that I said they're just starting this now. We have begun weekly aerial surveys in addition to our red tide surveys. They go on Thursday, so they go out tomorrow. And this is actually a red drum spawning aggregation from this year, just a couple weeks ago. I'm losing track of my weeks. It may have been last week. And there is probably 10,000 fish in this aggregation off Tampa Bay. Each of these individuals is 4 or 5 feet long. So you get a feel for the amount of biomass that is out there. We were happy to see this, frankly, because these red drum were aggregating and behaving normally. But unfortunately, we have multiple reports of large red drum dead, I would say in the hundreds. Not yet in the thousands, which would indicate an aggregation was hit particularly hard, but it's something we're keeping a very close eye on. The other thing that we've noticed this year is we've seen aggregations further inshore up the bay in Tampa Bay than we're used to seeing. For those of you familiar with Tampa Bay, the Sunshine Skyway, we've seen them on the other side of the Skyway, which we hope means that the aggregations are adapting to red tide being in the area. So we're hopeful there. The aerial surveys will continue and we'll get a good handle on impacts to those particular fish. Now, over the years, we've sampled these aggregations and we've tagged over 9,000 big bull red drum. So part of what we're trying to do is to get recaptures of these big bull red drum, and the tags are genetic. So if we get a fin clip, we'll be able to tell whether that was a fish we had tagged before genetically. And this is an area where we're enlisting charter captains. We're going to have kits for them very shortly that they can use to go out and sample fin clips if they see these big bull red drum dead, and that will allow us to do a marker capture and get a better feel for how red tide might be impacting these fish. Okay, thank you for indulging all of that detail, and this is where I turn it over to Jessica.